Hello, Sangar.
Hello, sir. Good morning.
Good morning, Sangar.
Hi, sir.
Ready?
Yes, sir. Yes, sir. I'm ready.
Okay.
Sir, shall we start, sir? Principal, sir?
Wait, sir. 9:50. 10 minutes.
No. Can I add them? Guest, sir?
We can add.
Okay. Add பண்ணுறதுக்கு
So I think I have joined here as a guest. Maybe can you give me the option to share the screen?
Yes, sir. I'll give that permission, sir.
Okay.
Sir, now we can try, sir. You can share your screen.
Okay, sir. Thank you.
Yes, thank you. Welcome, sir.
Can you see my screen, sir?
Yes, sir.
Okay, sir. Perfect. Hi, sir.
Hi, sir.
Sir, initially for some time we will share our screen, sir. After that introduction, you can share your screen,
Okay.
sir.
So I will stop now.
Yes, sir.
Sure.
തുടങ്ങിയില്ല
Good morning, one and all. Principal, sir, can we start the program, sir? Principal, sir, can we start the program? Sir, one Sir, can you start the program, sir?
Yes, sir, we can start.
Yes, thank you, sir. Selvasangar, sir?
Yes, sir. I'm here.
Yes. Thank you, sir. So, very good morning to one and all. On behalf of our chairman, sir, Sri M.N. Raju Garu, and our vice chairman, Sri Ravi Verma Mantanaharu, I wholeheartedly welcome all the principals of various colleges, staffs, and students of the various colleges for this wonderful webinar, the 10th series, what we are conducting in this series. So, once again, I wholeheartedly welcome on behalf of MNR College of Pharmacy to all the delegates for the 10th International Webinar on Salivary Microbe and its influence on diabetes and cardiovascular diseases by Dr. M. Selvasangar, postdoctoral fellow, Sidra Medicine, Kata. So I'm very happy to introduce MNR College of Pharmacy to the gatherings. MNR College of Pharmacy is established in the year 2004 and it is approved by AICTE and PCI. MNR College of Pharmacy is affiliated to Osmania University, Hyderabad, and we are offering courses like B.Pharm, PharmD, M.Pharm, and PharmD PB. We have a state-of-art pharmaceutical laboratories in the MNR College of Pharmacy, and we have more than 10,000 volumes of specialized books, and we have subscription for journals, and digital library facilities is also available for our staffs as well as student communities. Along with curricular activities in MNR College of Pharmacy, we encourage all the staffs and students to participate in various co-curricular activities such as conferences, guest lectures, and industrial visits. And we also encourage us to participate in extracurricular activities such as cultural and sports activities. More than 50 faculty members are working in MNR College of Pharmacy. Among them, 20 are with PhDs. The student team and faculty teams are guided by a well-known pharma field persons. There is none other than our principal, Dr. V. Alagarsamy, who is a recipient of Young Scientist Award from DST India, who is an editor of Anti-Infective Agents, a well-known Bentham Science journal. And he published on many reputed books. Among them, the most important one is Textbook of Medicinal Chemistry. Recently, he published on a textbook of Pharmaceutical Organic Chemistry, and it was available in that online also. And we have taken a special initiatives for coaching of university ranks, and we are providing a placement facilities, and we are giving coaching for higher studies, and we are guiding our students for foreign university admissions also. So our chairman sir will always tell that education and discipline is the two eyes. So hence in the MNR College of Pharmacy, we provide a favorable environment for studies, and we are offering education for the less caste, and we are motivating our students, and we are providing an SMS system for absentees and many more things. So with this brief introduction about MNR College of Pharmacy, now I am requesting the today's convener of international webinar and the principal of MNR College of Pharmacy, Dr. V. Alagarsamy sir, to give a welcome speech and to introduce the speaker to the gatherings.
Good morning, all the participants from various parts of the country, faculty members, principals, and my dear students. It's our pleasure to host this 10th International Webinar Series. We have reached this level of organizing 10 series of international webinars because of the enormous support from the participants. Every webinar, the number of participants have been increased. Sometimes it reached more than 2,500. That was the energetic force driven us to reach this 10th series of international webinar. So I thank all the participants for supporting us all the way. And while welcoming to this gathering, that is the 10th series of international webinar, So I thank our management chairman Sri MN Raju Garu and our vice chairman Sri Ravi Verma Garu and all our faculty members who are working day and night to make this program successful one. So I am proud to introduce the today's speaker, who is my student when I was doing in B Pharmacy. He is one of the best students who shows interest in the studies. Actually, so much interested in the chemistry, but later on, because he could get admission in the NIPER MS, so he has chosen this field of MS microbial pharmaceutical biotechnology. But still, the interest in the research is continued by him. That felt me very happy to call him for this international webinar. So Dr. M. Selvasankar, who is the Tamil Nadu Pharmacy Council member and Association of Mexican D Microbiology. He has more than 10 years of research experience in the same field of microbiology and biotechnology, and done B Pharmacy from MGR University under the SB College of Pharmacy, Sivakasi, and MS from NIPER, Hajipur. And he had obtained his doctorate from the Center for Research and Advanced Studies of the National Polytechnic Institute, Mexico. His area of research interest includes human gut microbiota and their association with the metabolic disorders such as obesity and type 2 diabetes. Dr. Selvasankar has many achievements and awards, and importantly, he has got all through thing because of this GPAT fellowship, followed by the European Molecular Biology Laboratory Award in German 2015. He has international and national publications, more than 10, which has highly peer-reviewed journals, and 7 chapters have been authored by him in the books. And he has attended many of the national and international conferences, and he served as a resource person, and he is one of the peer reviewer in the journals. And his research experience includes postdoctoral researcher in the Center for Research and Advanced Studies of the National Polytechnic Institute in Mexico, and currently he is pursuing the postdoctoral research associate in Sidra Medicine in the Translational Medicine Division, Qatar. So with these few words, I proudly welcome Dr. Selvasankar to take this session. Dr. Selvasankar, please.
Yeah, thank you so much, sir, for the grand introduction about me. I'm absolutely delighted to be part of this webinar. So I will directly start with my presentation. So I guess everyone can see my screen.
Yes, sir.
Okay. So the topic, today's topic, it's about one of the research interests from our lab, which is focusing on how the salivary microbiome and its influence, which is influencing in patients with diabetes and cardiovascular disease. This is part of the QNR project. QNR means it is the Kata National Research Fund Agency, which is focusing on personalized medicine, which can how we can program our health. So part of that program, it was funded to my PI, Dr. Sohaila Al-Qadar. So in this, the main aim is to identify whether is there any particular marker where we can focus, where we can utilize salivary microbiome as a marker to both for the diagnostics as well as the treatment. Before jumping to the today's talk, today the take-home message will be, at least you will have some idea what is microbiome and how this microbiome is involving in our health. So today's talk, the content will be introduction, the main justification of this project, aims, experimental strategy, methods, results, and conclusion. Before to dive into the talk, I want to give a brief a story how this journey of microbiome started all over the world. A field of this microbiome started since 17th century. Most of us, we have heard about this famous scientist, Anthony von Leeuwenhoek. in the period of 17th century. During his period, he was not even considered as a scientist because basically he is not a qualified person to study biology. His profession is just to make glasses. So his family dedicated to that, but because of his curiosity, he was making some magnifying glasses and lens. Through that lens, he started to see the different things. So one day, one part of his curiosity, he just take a sample of his some dental plates, and using that magnifying lens, he started to see some moving creatures. That day, he started to modify in his lens, and he started to see different creatures inside his dental molecules. In that, here he whatever he found during that time using a single lens, he just draw the diagram and he forward that to London Real Society, where the scientists earlier they didn't believe the existence of this undiscovered universe in the world. So later he with all his proofs, they also tried the same. After that, they confirmed the existence of this new universe, where he started to later they call that molecules as bacteria. So he's the father of microbiology. So here in that creature, curiously, this come from the saliva, from the oral microbiota. So I can, by the structure, I can say that this is this stretched molecule. This bacteria is spirochetes, and boomerang shaped this bacterial molecule. This is leptotrichia, and these are definitely it will be streptococci, which are the most common microorganism which exist in the human saliva. And flagellal type, this movement, it's possibly due to Selenomonas, which is the one and only bacteria which has the oral bacteria which has flagella. So with this, what we can conclude? Yes, there is always these microorganisms which exist along with our human body. It doesn't involve directly in the disease. It always living in a symbiotic relationship with the human host. With that, for the today's talk, we will be using frequently these terms. So I want to give a brief definition of the microbiota, microbiome, and dysbiosis. What is microbiota? Whatever the microorganisms which resides in our human body, which includes bacteria, archaea, viruses, fungi, and several other eukaryotic molecules which lives inside the human body, they are considered to be microbiota. And microbiome, the collection of all genes which is encoding by this microbiota, are called as microbiome. When there is an imbalance between the diversity and the abundance of this particular microbiota, that we will call as dysbiosis. So with this, I want to give a brief introduction about human microbiome. We are more microbial cells than human cells. In our body, we have 10 folds higher of microorganisms than human cells, and these microorganisms, they are encoding for the genes which is 100 times more than our human genes. So our human body and this human microbiome, they are in a very good relationship, which is involving in almost all our physiological functions. So our journey with the microbiome, it started even when we are in the placenta. So earlier it was considered to be the placenta is sterile, but fewer studies in the earlier years explained that the placenta is not sterile. There itself, there is a translocation of microbiome between the mother and the baby. But still there is several conflict, but we will not focus into that. But definitely we are getting in contact with the microorganisms during the time of delivery, either with the C-section or the normal delivery. That microorganisms helping us to develop our immune system. Over the time period, the diversity of the new baby microorganisms is changing due to several factors, which involves many environmental factors, during our life, whatever the diet we take, our regular activity, and any antibiotics treatment, or if anyone has pet, based on that, our microbial diversity will affect. So there is a difference between the diversity of those who lives in a rural area and those who lives in an urban area with restricted lifestyle. So the diversity is completely different. So when you are exposed to different types of antigens, whether it comes from diet or from any microorganisms, that will help us to develop our immune system. And since these microorganisms, one of the important thing to modulate our immunity. So when we have a good diverse microbiome, we are in a very good healthy shape. When that particular diversity affects, when there is an imbalance happens, that will lead to cause certain diseases. In the same way, when we go from younger to older, our diversity start to reduce. There it will cause several consequences which affects our immunity as well. So still there are many factors, even the microorganisms which are involving in several brain-related functions. Still there are many studies going on. So in this, further in human microbiome, there are many studies that explain every human being, they have their own microbial diversity, like a personal microbial cloud. Every person has different microbial cloud, and inside the human body itself, based on the different body size, the diversity changes. So here you can see the different body size, like the mouth, our gut, and then the other skin and the other body sites, which has different diversity. Among all body sites, gut microbiome is the most diverse microorganisms. Next to similar like gut microbiome, oral microbiome also most diverse. So the wall is the root where we are sometimes through inhaling and other normal environmental exposure, we are getting exposure to the new microorganisms. So here you can see there are different microorganisms. Just a slide where we can see the diversity in the microorganisms. So here the further explanation how the microbial composition it's affecting based on different factors, like our regular diet, whatever our environmental factors, and several genetical factors, and other microorganisms, other microRNAs during over the period of age and our lifestyle, which can affect the microorganisms. When there is a change in this microbial diversity, there it will its consequences can cause several problems. So in Sidra, part of our group, we are mainly focusing on to study human microbiome based studies in different diseases. So part of this, we have a well established lab in our institute facility. So once we receive the human samples from the different body sites, our aim is to, it depends upon the focus of our project. If we want to characterize the profiles of the microorganisms just to extract the total DNA, then we either using 16S DNA based sequencing or the shotgun sequencing, we can characterize the community profile. If you want to see the gene expression level, in that case, we will extract the total messenger RNA, then we will do the meta-transcriptomics. If you want to focus the human microbiome gene expression levels, then the term we will use is meta-transcriptomics. If you want to see what are the proteins that can be expressed by this human microbiome, for that we will use metaproteomics technology using gas chromatography and other mass spectrometric techniques to study the metabolites which are produced by the human microbiome. For that, we use metabolomics technology using the gas and liquid chromatography. So in this study, we focused on 16S RNA-based diversity identification. Why we choose next generation sequencing? Why we didn't do the normal culturing techniques? The difference is most of the human microbiome, they have diverse microorganisms most of the microorganisms, they are not culturable. So that's why due to the development in the late 2000, around 2008 to 2010, there was a huge development in the sequencing technology, which helps to from the extracted total DNA, we can, using the sequencing technology, we can identify the microorganism without the need of culturing them. So in that next generation sequencing technology, the good facility is at the same time, you can process hundreds of samples in a single sequencing, which is better than the earlier Sanger sequencing technique. So in this, the difference between metagenomic sequencing and 6NS1, from the total extracted DNA, this 6NS base, it's a target specific amplification. Once we do the target gene amplification, from that amplified library, we can identify the microbial diversity. In the shotgun sequencing, the total DNA will be shared into many fragments. Then the fragments will be sequenced, and then they will be assembled again as a contigs. From that, you can identify not only microbial diversity, you can also recognize their metabolic routes as well. This shotgun sequencing, it's more expensive, but it will give you more depth information about human microbiome. So in this study, we are focusing why we are interested in diabetes and cardiovascular diseases. But as a non-communicable disease, diabetes, CVD, and cancer, which accounts for more than 70% of death around the world, diabetes can cause around 1.5 million deaths every year. It's a multifactorial disease which its further consequences causes several health complications like hypoglycemia, retinopathy, nephropathy, and neuropathy. And cardiovascular disease, it consists several heart-related problems, including coronary heart disease, heart failure, hypertension, stroke, arterial fibrillation, and cardiac death. It is one of the leading cause of death around the world, which is causing 79 million deaths. The justification of this project in Qatar, the most common people which affects the health of the Qatari adults is type 2 diabetes. So in this, we want to take saliva as a focus of sample. So since the saliva, it has many kind of nutrients, many electrolytes, many protein molecules, and several microorganisms. And it is very easy to collect and non-invasive. When there are earlier reports in other population has been reported, when there is a dysbiosis happen in the oral microbiome, it can cause several metabolic diseases like obesity, type 2 diabetes, and other diseases as well, cancer, rheumatoid arthritis, Parkinson's disease. So there is no earlier previously reported systematic study which can explain the role of salivary microbiome and how it's affecting in type 2 diabetes and cardiovascular disease in Qatari population. So the aims in our project, first to characterize the salivary microbiome composition in Qatari adults with who has cardiovascular disease risk and type 2 diabetes, and to identify any particular microbial signature which can explain their role in those who are with diabetes and cardiovascular risk. So the panorama of this project, now Qatar, they are working on a project which is called Kata Genome Project to characterize the whole genome of Qatari population. So part of that program, we collected samples from 1,000 Qatari genome participants. So from that, we collected their clinical data and their saliva samples. So from the collected saliva samples and the clinical data, once we characterize the salivary microbiome profile using the multivariate analysis, we will look for any model which can explain any particular salivary microorganism which can involve in the diseases. So for this study, since we are focusing on diabetes and CVD risk, how we are scrutinizing the groups in this study. for diabetes using their HbA1c, which is the glycosylated hemoglobin level. Based on their level, we are classifying the participants into 3 categories. If the HbA1c level is less than 0.7, they are considered as non-diabetic. If it is between 5.7 to 6.5, they are belonging to pre-diabetic group. If they are above 6.5, they are considered as diabetic category. For CVD risk calculation, we use Framingham risk calculation formula, which involves, in the next slide, I will explain what are the parameters we took into consideration to calculate this risk score. So based on the risk score, we are dividing them into 3 categories. If the score is less than 10, they are considered with CVD low risk. If the score is between 10 to 20, they are considered as intermediate. If the score is above 20, in the next 10 years, this is just a prediction, in the next 10 years, they have the higher possibility to have cardiovascular diseases. So these are the parameters which we include to calculate this CVD risk score. So based on the gender, age, and the total cholesterol level, HDL cholesterol, and their BP level, and their HbA1c level, using that, we can predict the cardiovascular disease risk. And for the other method which we use for this project, most of the participants, the audience here, they might have know about this technique, but most of them are students who are in the bachelor degree. Maybe for them, I'm going to explain what is polymerase chain reaction. This is one of the basic molecular biology technique. So in this, if we want to amplify our target gene, so we need our target DNA. From that, we have to design our gene of interest primers for our gene of interest, which is called sense primer and antisense primer, or forward and reverse primer. Then in the PCR, there are 3 important steps. One is denaturation, annealing, and elongation. During the denaturation, our target DNA, which is double-stranded, it will be separating into single-stranded DNA, and then using the annealing temperature, our target primer will bind into our target DNA in the both forward and reverse side. Using the tag polymerase and the DNTPs, it will start to amplify in the direction of our gene of interest. Then, like that, in several cycles, this process is repeating, so we can amplify more of our gene of interest. Using that strategy for this project, we have collected the samples simply by spitting the saliva, and those saliva, we extracted the total genomic DNA. From the extracted DNA, here our gene of interest is 6' ribosomal RNA. This 6' ribosomal RNA has 9 different genes, which is called hypervariable regions. This hypervariable regions is like a how the fingerprint for human beings. Like that, this hypervariable region is unique for each bacteria. So in this project, in this work, we have targeted the region V1 to V3. Once we amplify that product using the PCR, during the PCR, in our primer itself, we have added a barcode sequences, which is 10 to 12 base pair sequences. That sequences, which is different, unique for each sample. So in that way, we amplify our samples, each sample with different barcodes. Once we have the product, that will be purified, and then we can mix all those products in a single tube, which is called massive pool preparation. Then the mixed sample will be sequenced through the next generation sequencer. This is Illumina. So after sequencing, using that particular barcode sequences, you can extract the sequences for each sample. Then we will check the quality, and we will go for the bioinformatic analysis to check the profiles in the salivary microbiome. So this is the steps which involves during the data analysis. First, we will check the quality, and then using the KIME tool, we check the microbiome profile. one step is to identify the microbiome profile. The next one will be alpha and beta diversity. This is to check how the diversity changes among the samples and groups. And finally, we will do the correlation analysis to check what are the clinical parameters which can be which is possibly correlated with the certain microorganisms. We use two different tools, HALA and networking tools. In the later part of the slides, I will explain how this analysis will work. So here the clinical table for the CVD group. So based on the CVD score, using the formula as I told earlier, most of those who are above the score, when the score is above 20, they are considered as high risk CVD group. When the score is between 10 to 20, they are intermediate. When the score is lesser than 10, they are low risk. So here the other parameters, insulin, glucose, HDL, LDL, cholesterol, and triglycerides. So totally in this study, earlier we collected 1,000 samples. Among them, few samples we got inadequate amount, so we have excluded those samples from our study. So totally we end up with 995 samples, 441 are male and 554 are females. All the people are we didn't apply any particular exclusion criteria, and the inclusion criteria just everyone should be Qataris. Before to move into the results, just most of us we know what is this taxonomy. So it's just to when we describe our result, just to have some idea, I put this slide. So how the taxonomy works, it's in the order like same way: kingdom, phylum, class. After that, order, family, and genus and species. To reach the species level, we have to do either shortcut sequencing or the whole 16S ribosomal RNA. So in this, we just amplified only the gene B12V3, where we can reach only till genus level. So first, I want to give a overall profile how the salivary microbiome in Qatari population, how it look like. When we analyze the data at the phylum level, the most common phylums here are bacteroidetes, firmicutes, and proteobacteria. The most common we found 72% of bacteroidetes. When we focus at genus level, the most common bacteria are Prevotella, Porphyromonas, and Streptococcus. After that, Legionella. So these are the most common members in Qatari salivary microbiome. Then we wanted to compare whether this Qatari salivary microbiome, how they are looking when we compare with the population from other countries. So we did a comparison with the countries like Brazil, Bangladesh, USA, Korea, Germany, UK, and Japan. So here you can see there is a difference in the profile in Qatari population compared to other country population. So the most common member, the most top abundant member here is the bacteroidetes, and next firmicutes, and then proteobacteria. Then we did a beta. Okay, before to explain about how the diversity works, the two important parameters in the diversity analysis here are alpha diversity and beta diversity. Alpha diversity. Here, how many different types of sequences are available in sample? That means how many different bacteria exist in a particular sample. That explains alpha diversity. Beta diversity, when the samples, how when they are you are clustering in them into different groups, how the samples in the group they are different with the other group. To compare between groups, that analysis we call as beta diversity in a simple words. So here we compare the beta diversity of the salivary microbiome of the Qataris with other populations. Here you can see the Qatari population salivary microbiome, it's clustering completely different with other nationals, so it is significantly different. Early this year we published this work, which is available in Journal of Translational Medicine. So next we will move into how the microbiome profile is different in cardiovascular disease risk group. So here you can compare at phylum level how the diversity changes. Here you can see there is a slight changes in bacteroidetes level from low risk to high risk, there is a slight decrease in the bacterial level. But on the other hand, there is an increase in firmicutes level in compared to the low risk. There's a decrease in firmicutes in low risk compared to intermediate and high risk group. When we focus into genus level, there is a slight decrease in Porphyromonas in compared to the intermediate and high risk group. On the other hand, Prevotella is significantly higher in low risk compared to intermediate and high risk group. So here you can see another analysis called Lepsis analysis, which is the full form is linear discriminant analysis, where this analysis will apply to the whole microbiome profile to identify what are the marker microorganisms which are significantly abundant in one group compared to the other group. So these are the green bars which are significantly higher in low risk group, and the red bars are the bacteria which are higher in high risk group. To identify the alpha diversity, we used 4 parameters: observed species, Chow1, and Shannon and Simpson index. In this, these 2 parameters are the parameters which we use to calculate the species richness, how many different members. And Shannon and Simpson are the diversity indexes. But here, unfortunately, we didn't find any significant difference among these groups. And we didn't find what we expected using the beta diversity. There will be a completely different profile between low risk and high risk group. But here we didn't see that kind of particular cluster. So the profiles almost look like same, but there is a change in certain microorganisms and in their abundance level. We didn't find a significant difference. Then to identify using the co-networking analysis, co-networking is whether there is a particular relationship between within the microorganisms, any particular bacteria, they are in a mutual relationship or they are in a negative relationship with any other bacteria. To identify that, we did the networking analysis. Using that, here you can see those connection with blue color, they are negatively correlated, and the red one are positively correlated. Here you can see bacteroidetes at in low risk and the high risk, they are negatively correlated with proteobacteria and firmicutes, which what it says, when bacteroids are increasing, the abundance of firmicutes and proteobacteria will reduce. In the same order, at genus level, you can see Prevotella is the vital bacteria which negatively correlated with bacteria like Streptococcus, Legionella, Granuliculata, and Neisseria. So vice versa, these bacteria are negatively correlated with Prevotella. Same way we compared with high risk and low risk and moderate risk. So same you can see bacteroidetes, they are negatively correlated with firmicutes and proteobacteria. In the same like the previous section, Prevotella is negatively correlated with Streptococcus, Hemella, Haemophilus, and Neisseria. Then to identify the prediction technology, we used random forest analysis. In that, we predicted the Prevotella. Prevotella is one of the factors which is significantly higher in high risk, low risk group compared to high risk, and in high risk, Tannerella was the bacteria at genus level which came significantly higher in high risk group. So these 2 bacteria can be a marker to predict type 2 cardiovascular disease in the participants, but further analysis need to be done at both in vitro and in vivo model. In the same way, we compared using low risk and moderate risk. In that, you can see Prevotella again, one of the high significant marker which came in low risk, and in intermediate, we found Streptococcus. Then we did the correlation analysis between whatever the blood parameters like the glucose level, lipid level, triglycerides, and albumin level, all the molecules which we can study in blood samples. So what is this analysis? The analysis we call HALA. HALA means an association between all against all the microbiome profile against all the available metadata. So in this picture, what you see here, when there is a significant correlation between microorganism and the blood parameters, there it will create that number. The numbers are ordered in their significant p-values. So those who are numbered in red color, they are positively correlated. Whatever the numbers which is in blue color, they are negatively correlated. Here you can see the correlation score range. So from this you can see leptotrichia and mycoplasma, which is positively correlated with creatinine and uric acid. Like that, Veganella, it's negatively correlated with urea. Streptococcus, it's positively correlated with C peptides. Same way, we did the correlation with the group, whatever the samples from intermediate group with the blood parameters. Here we found Megaspira, which is positively correlated with total cholesterol level, and leptotrichia as well positively correlated with cholesterol level, and Porphyromonas is negatively correlated with calcium level. Same way we found with high risk group, that Rothia, which is positively correlated with insulin level. So that means when insulin level increases, this Rothia abundance also increases. So this can be one of the target bacteria which can explain the risk, cardiovascular risk in Qatari participants. So with this analysis, we will start with the our next part of study, which is diabetes. As I explained earlier, so in diabetic group, we divide them into 3 categories: non-diabetic, pre-diabetic, and diabetic. In diabetic, so using the HbA1c level, when it is less than 0.7, they are non-diabetic. When they are the HbA1c level between 5.7 to 6.5, then they will be considered as pre-diabetic group. When it is above 6.5, they are diabetic. So the other parameters which we included here, so same group of sample, we just compared them for the diabetic profiles. So here you can see the abundance of at phylum level, you can see the abundance of bacteroidetes, which is lower in compared to the pre-diabetic and diabetic category, and there is no big difference at firmicutes level. At genus level, you can see there is a difference in prevotal abundance compared to pre-diabetic and diabetic. Same like here, the Streptococcus level is slightly increased in non-diabetic compared to the diabetic and pre-diabetic category. Same like CVD group, here we didn't find a significant difference among the diversity. We're using the parameters which we have calculated, observed Chow-1, Shannon, and Simpson indices. But at the beta diversity level, so here you cannot see, but at the 2D level, you can see there is a significant distance among the microbial members community from each. Here, each dot which indicates the microbial community of each sample. If it is orange, they are diabetic category. If it is blue, they are normal. So we compare between both normal versus diabetic and pre-diabetic versus normal. So in this, we can see there is a slight difference in the community between normal and diabetic category. So with that, we did the correlation network analysis to compare the profile. As we expected, here bacteroidetes is negatively correlated with Firmicutes and Proteobacteria and Actinobacteria. Same, Prevotella, which is the vital factor, which is negatively correlated with many bacteria like Veganella, Hemella, Neisseria, and this Veganella, which is positively correlated in diabetic group with other bacteria. So as I explained earlier, here if it is the blue color, then they are negatively correlated. The red one are positively correlated. Again, by comparing between non-diabetic and pre-diabetic group, we got the same. This Veganella, which is positively correlated with several bacteria like Streptococcus, Atopobium, Haemophilus, Hemella, and Prevotella is negatively correlated with other bacteria like Streptococcus, Veganella, and Neisseria. When there is an increase in Prevotella, then there will be a decrease in diabetes level. When in the diabetic group, there is an increase in Veganella abundance. So using the random forest prediction model, we found that this Veganella is significantly higher in diabetic group compared to the non-diabetic. Same vice versa, we found Prevotella, which is significantly higher in non-diabetic compared to the diabetic group. But same comparison with non-diabetic and pre-diabetic group, we found that Veganella, which is significant even as in pre-diabetic as well. So this Veganella can be a prediction marker to use to predict diabetes in this study. We did earlier, like in the CVT group, we did the correlation analysis using the blood parameters and the salivary microbiome profile based on the category. So here we found Streptococcus is significantly negatively correlated with albumin and creatinine, and other bacteria like Tannerella and Campylobacter, which is positively correlated in participants with no diabetes. We did the correlation with the pre-diabetic group. Here we found Streptococcus is positively correlated with insulin, C-peptide, and glucose level, and Veganella is negatively correlated with potassium level. In diabetic group, we found Veganella, which is positively correlated with glucose and HbA1c level. Earlier also we saw in the networking analysis, this Veganella is one of the important marker bacteria which we found significantly higher in diabetic group, and also they are positively correlated with the other bacteria which is significantly higher in diabetic groups. Here you can see its positive correlation with the important parameters which are related to the diabetes like glucose and HbA1c level. So with this, we predict that this Veganella can be a predictor to predict diabetes. So another part of interest in this project, which is to identify whether, apart from the microbiome profile, we wanted to compare the salivary microbiome proteomic profile with the plasma profile. The idea behind this aim is to compare whether saliva can reflect the same protein profile, same like plasma profile. So if it is so, we can use saliva as a sample to diagnostic purpose instead of collecting the blood sample, which is little invasive. Little invasive to the patients, and many people, they are not interested to give blood sample for their prediction analysis. So in that idea, we use the SomaScan technology. This is one of the important advanced technology where you can use many samples. You can process many samples at a time, and they are highly sensitive and quantitative and reproductive tool. So in this tool, you can identify many biomarkers you can use for drug discovery, preclinical and clinical analysis. You can analyze up to more than 1,300 protein molecules in a very, very small volume of sample, like 150 microliter. You can process any kind of biological sample like serum, plasma, spinal fluid, and also other biological fluids like saliva. So as a part of this project, so we are in the state, so we wanted to compare. So we collected plasma and saliva samples from the 12 different volunteers to check whether we can able to see the proteomic profile between whatever the proteomic profile we obtained from plasma in same sample of saliva. So here you can see, even though the abundance level is different, but we can able to identify the same proteomic profile. Whatever the profiles are we saw in a plasma sample, it can reflect the same in saliva sample. With that, we identified for the diabetic group. In this study, we identified the proteomic profile. We almost got the profiles for more than 1,340 samples, 40 different proteomic profile. Among them, these are the few protein molecules which we found significantly higher in non-diabetic group, and few markers which are significantly higher in diabetic group. We are in the stage where to integrate this proteomic profile with the microbiome data to identify any particular marker, particular signature, which can explain the role of salivary microbiome and diabetes. So with this, I want to conclude today's talk. This study is one of the first study to characterize the salivary microbiome in Arab population, and one of the study which involves a huge number of samples, more than around 1,000 samples. Our data shows that the salivary microbiome profile, it's completely, Qatari population is completely different to other nationals like Brazil, Japan, South Korea, Germany, UK, and USA. And from this correlation analysis, we can identify that Veganella is one of the important significant marker, which is significantly correlated with glucose and HbA1c level. To further study, we need to process the samples by studying meta-transcriptomics and metabolomics analysis to identify their expression level and their metabolic routes. With that can give a more deeper knowledge to understand their development in diabetes and cardiovascular diseases. So the end of this talk, I want to conclude. I want to acknowledge all our team members and our collaborators and all the participants who involved in this project. I would like to thank all the participants. I look forward to your questions. Thank you so much.
Yes, thank you, sir, for your wonderful presentations on salivary microbiome. Yes, participants, if you have any queries, you can raise your hands. I will unmute yourself and you can ask your questions. Any participants, if you have any queries, you can put in the chat box, or otherwise you can raise your hands. That I will unmute yourself, that you can ask your questions. And one of our participants, sir,
Yes.
that just a minute. Is it possible to perform this particular studies in India?
Yes, there are many studies already. It has been published in India. Many different labs, they are working on human microbiome studies. The thing is, for sequencing, it is a little expensive. Most of the government institutes in Pune, I think one institute, the National Cell Laboratory, they are working on the human microbiome projects. And even in many small labs, also they are working on that. Recently, there is a development of a simple sequencer called Axport Nanopore, which is like a benchtop sequencer. Anyone can buy that sequencer and they can perform the sequencing.
Thank you, sir. And another one of the candidates, they are asked in that YouTube candidates, they are asked, what is the salivary microbiomes present in Indian populations?
So overall, in many populations, it is varying between the country to country. But normally, the top abundant member, which we can call them like the core member of salivary microbiome, is Prevotella, Porphyromonas, Streptococcus. these are the common in all over the world, but in Indian population, there is a higher number of Streptococcus compared to the other bacteria. In Qatari group, we found more Prevotella than Streptococcus. In Indian population, we found more Streptococcus.
Thank you, sir. And one of the other candidates asked, what is the reasons or the mechanism behind how these microbiome alters these diseases? Whether that microbiome undergoing any changes or otherwise it is altering any metabolic process in the human beings.
So it's a tricky question, very good question. So it's like same like one question like who came first, whether the egg or the chicken? So that is we exactly don't know the reason whether due to change in our human physiology that affects the microbial diversity or because of the change in the microbial diversity which is affecting the human health. So that factor which is the one everyone we are focusing to understand. So there are several animal models and in vitro models people are working, but still there is no clear answer for that. So in this work, just we are giving a panoramic view how the profile is changing in different disease conditions.
Yes, thank you, sir. One of the another delegates asked, can the salivary microbiome balance can be corrected?
The idea, there are several technology people are suggesting. Earlier, they suggested just by using any probiotics or any prebiotic diet, you can alter the gut microbiota. And even recently, they suggested fecal microbial transplant. In that, those who have any severe effect in the dysbiotic microbiota, you can get the donor, healthy donor, you can transplant the good fecal diverse microbial transplant to the health, those who has problem, then you can balance it. But still, there are several clinical trials are going on, and recent times there are also development of CRISPR technology where you can, it's like programming, you can modulate, you can program what kind of bacteria you want to have in your gut microbiome. But still, they are working in animal models. Maybe in future they can come up with a very good answers for this.
Yes, thank you, sir. And in your previous answers, you told about on animal microbiomes. And the next question from the delegate is regarding that animal microbiome only, sir. Is it possible to use that animal microbiome for the studies?
Yes, but depends upon what kind of studies they want to study. So in how they in earlier, when someone come up with the idea to focus on human microbiome, which is involving in the health. So at the initial point, nobody trusted that whether the human microbiome, which is involving in the health. So what they did a comparison of this germ-free mice, which is completely free from organisms. So they transplanted human microbiome to those animal models. That causes many, for example, those people, obese people, they collected the microorganism, they transplant that to lean mice model. After receiving that microbial transplant, that lean mice, they transform to obese mice. That indicates there is a significant role of these microorganisms which can affect the metabolism.
Yes. Thank you, sir. And another question from our delegates. Is it possible we can grow that microbiome and transfer to the patients, and from that can we use this microbiome for treatment of any diseases?
So the idea behind this project, as I told, we cannot culture all the bacteria. That is the problem which we are facing in this. So once from this analysis, once we come up with any particular significant marker bacteria, then we can focus on that growing of that particular bacteria. And first we have to work on any animal models. After that only we can focus on that. We cannot directly suggest to the patients.
Okay, thank you, sir. And there is a common question, sir. As you are working in Kata, And what are all the opportunities? Because most of the participants, those who are participated here is UG and PG students. So what are all the opportunities for our B pharmacy and M pharmacy candidates in the Qatar, sir, and what they have to do? If you have any overview, if you are telling that things, it may be helpful for our young pharmacists, sir.
So from my experience, there are many people who has pharmacy background. They are working here as the hospital pharmacist. Apart from that, Qatar, they have their own pharma company. They can also look for the opportunities there. And for the masters and for further research career also, Qatar has many research institutes where they can focus their studies. If simply with the B Pharm, then the option here in Qatar, it will be hospital pharmacy.
Okay. Is there any specific exams, or if you want to come in that Qatar as a pharmacist, or if you want to do our postdoctor fellowships, or if you want to do our PhD for those things, any specific exam is there?
For PhD and postdoc, you don't have to give any exams. Just you need, based on your records, you can get your job. But for the pharmacy, yes, like other Gulf countries, you have to obtain your license. For your license, you have to give exams.
Thank you, sir. Thank you for your spending your valuable time, sir. And that we got a feedback from most of the delegates that the session was very interesting, and we learned on something newly, like that many delegates, they had given their comments in the YouTube as well as in that Zoom chat box. They are praising that the presentation was very neat and it was on simply superb like that. And thank you for your wonderful presentation, sir. And thank you for spending your valuable time and giving an explanation for all the queries what it was asked by our delegates in the Zoom as well as in the YouTube. So now yes, thank you, sir.
Yeah. Yes, sir.
So now I'm requesting our principal, sir, to conclude these sessions. Sir Selvasangar, sir, can you stop sharing your screen, sir?
Yeah.
Dr. Selvasangar,
Yes, sir.
just as a pharmacist, your view for the who are doing undergraduate or postgraduates, in where they can enter in the biotechnology or microbiology related higher studies, because one of the area where you have entered is it's a unique. Till now, drug design, pharmacological screening, all these are all one approach for the any diseases. What you are doing is one of the typical study that we could understand. So like similar type of things, similar studies, the microbiology or biotechnology related areas, where the pharmacist, that is, our Indian farmers, B pharmacy or M pharmacy students, what type of research work they can carry out in the abroad. Can you suggest something on this?
So if we focus on the biotechnology related field, so I can suggest there are in the recent times many pharmaceutical industry, they are focusing on the human microbiome based studies. So if they in US, in Mexico, in several, even in India, several pharma companies, they are focusing on microbial instead of microbial transplant. Earlier, there are many clinical trials are going on which focus on the fecal microbial pills, which can to make to stabilize the human gut microbiome. And for the bachelor's and master degree students, yes, they can, if they are interested in this biotech related field. So yes, they have huge opportunities in the coming years when it comes to the market. In Mexico and other countries, there are many, those who are interested in research, there are many scientists who are willing to take students. There are many scientists there are more huge opportunities with the funding options, but there are lack of students to get who are interested to do the research work there. So for the Indian students who are looking for the opportunity, I can suggest that instead of focusing on only the developed countries like US, UK, you can also go to these countries where they have a very good established research structure. We can focus on that.
So thank you, Dr. Selva Shankar, because nowadays the population is increasing, the number of avenues also increasing, but there are no people to say for the young graduates. So they feel that opportunities are confining or opportunities are shortening, but that is not the fact. By seeing you, we can see there are so many avenues, even in the biotechnology related, even the drug development or your area of type of research also, our pharmacy students can enter. So this is one of the good light that you have thrown to our that is pharma graduates. So hope many of the students and the faculty members who are attended, please convey this thing to your students, because the intention of calling Dr. Selva Shankar for this webinar is, it's not that he is my student. One thing, he is my student, he can deliver well, with that I called. Second thing, he definitely this type of new topics will throw some light on the students who are feeling that pharmacy profession, what next? Especially some of the department, they say they don't know what is the next opportunities. So for those type of students, it will be very much helpful. With this mindset only, we always arrange the speakers. We in our earlier seminar webinars also, we can see that the speakers all we got from, they are all excellent in their own field. So each one have contributed, or each one have delivered very well to the audience. So all the audience also told that they have taken something to their home as a take home message, and everyone got very good inputs from the webinars that we have organized. In the same way, today Dr. Selva Shankar also very much justified to the audience. I thank Dr. Selva Shankar on behalf of the organizing team and our management, our chairman Sri MN Raju sir, and our vice chairman Sri Ravi Verma garu. So kindly accept all our thanks to Dr. Selva Shankar, and definitely in coming days also you can do such seminars not only for our college, in our for our Indian students, so that they feel happy or they feel new avenues for their career. So with these few words, I thank Dr. Selva Shankar. So we would like to present a small certificate as a token of our love and appreciation to you. Kindly accept this virtual certificate. It's like really I feel proud to hand over this certificate. Still I remember the first year B Pharmacy, where I taught you organic chemistry, and you also spoken very intimately about the subject. Leave about the personal things. Only always as a teacher, we always remember what type of subject interaction we had. So your that curiosity that was in the organic chemistry, it has grown up very well, and now that's why you are here. Still, all your chemistry knowledge also you can put along with your microbiology, so that you can do wonders in the coming days. Because all the basic thing that we need is the curiosity that you have very well with you. So hope with these inputs, definitely you will also do well for the suffering community, especially present world is suffering with microbes only. So more than the microbes only is affecting us, but you are telling microbiota only helping us. So if this coronavirus including us indirectly, if any of the humanity or any of the things that is missing, if it is brought back, that may be good news for our humankind. So with these few words, I thank all the participants, and we would like to inform you that with this, we are concluding the international webinar series 10th. So with this 10th series, we got a huge response, and we have got so much energy to carry out the further programs. Hence, as per the many of the students' request and many of the faculty request also, we would like to organize after a 1 week of gap. We would like to organize a series of webinars based on the title of, we got a huge response and we got opportunities for the pharma graduates, various opportunities for the pharma graduates. In this title, we would like to invite the eminence from the various departments who are doing excel in their field. Like one vice chancellor is going to address, another one successful academician, then one journal editor who is one of the senior that from whom I learned, and many of the marketing people. So marketing can be taken as a career. Then in the that is clinical pharmacy, and in the pharmacovigilance, this can be taken as one career. So like this, we have taken 10 different areas for the webinars. So while thanking this webinar series, I also request all the participants to join, and also you spread the message to your students also to join in this, so that they will feel encouraged, or they will feel happy about the pharmacy profession by listening all these career guidance type of webinars. So with these few words, I thank one and all for joining us. Thank you very much. Thanks a lot.
Thank you, sir.
Thank you, sir.
For the nice opportunity.
Thank you, sir. Thank you one and all for your participation in this international webinar series. And the feedback link will be activated within 5 minutes. You can send your feedback, and from that you can obtain your, for getting a certificate, it is feedback is must. So already feedback form is activated, and many people you are asking that only. Feedback form is activated, you can submit your feedback. Yes, thank you, sir.
Any participant before leaving, any participant who would like to comment means we are most welcome. Maybe Dr. Professor Balvin Singh, so you can say, or any of the professors or faculty members, if you want to have any comment on our webinar series, so you are most welcome.
Yes. If you are raising your hands, I will unmute your unmute. You can talk, sir. Anybody, if you want to give your opinions, you can raise your hands. I will unmute you. Then you can share your opinions.
Professor Balvin Singh, Dr. Saravanan.
Sir, yes, sir.
I think you can unmute him.
Yes, sir. Yes, sir. I will unmute, sir. Sir, please unmute yourself, sir. I already given permission, sir. Professor Balvinder Singh, sir. Sir, please unmute yourself and you can talk, sir.
Okay, if nobody else, if any other faculty members, would you like to comment or would you like to say anything, means you are most welcome.
Feedback link போட
Right, sir. Saravanan, sir. With this, I think
Sir,
we can conclude.
Some of that student, they raised the hand, sir. I will ask.
Okay.
Yes, Segu Pratyusa. Please unmute yourself and you can talk.
Hi, good morning, sir. I'm from Lepsis College of Pharmacy. Very excellent sessions. The session. And what are the publications we can publish articles like?
Hello.
Are you getting?
Your voice was not clear, madam. Please.
Hello.
Madam, tell me, madam.
Excellent. What's the main object of topics? Hello.
Madam, we are not getting clear, madam. Please.
I think signal is not clear here, sir.
Okay. Tell me, madam. Now it was clear.
Sir, it is excellent, sir. I'm at Oxford College of Pharmacy, Bangalore.
Yes.
What is the main conclusion of the topic, sir?
So madam is asking, she's from Oxford College of Pharmacy, Bangalore. She's asking about what is the conclusions that we are giving from this particular topic. So I think the question for Sangareddy, sir. Sir, please, Sangareddy, sir, please unmute yourself and you can talk, sir.
Yeah.
Yes, sir, now I guess.
The main aim of this project is to identify whether we can identify any particular marker bacteria which can predict or can be used as a probiotic in the future to treat diabetes or the cardiovascular disease. We are in the process to identify that particular marker. So once we identify any particular marker bacteria, the next stage will be we will do any kind of cell models. After that, we will go with the animal model studies to confirm whether really that bacteria is involving in the physiology of human cells. Using any animal model study, that is our next aim of this project. At the moment, we are in the prediction of any marker bacteria which can use for diagnostic or prognostic purpose. Thank you.
Okay. Okay, thank you, sir. Thank you very much for your conclusion.
Yes, thank you, madam.
I think we can conclude, Saravanan sir, with this.
Yes.
Okay. So thank you all for joining. Thank you, Dr. Selvasangar, and all the participants and all the organizing committee members also. Thank you, one and all.
Thank you so much, sir.
Saravanan, you can sign off, sir.
Yes, sir. Thank you, sir.